Welcome back. We will be discussing chapter number 2, that is sexual reproduction in flowering plants. Flowering plants are angiosperms, whereas in gymnosperms they do not produce any flowers. Sexual reproduction is present. So we will restrict our. You already know that the sexually reproductive part in a plant is its flower. So the phase of sexual reproduction in plants can be divided into 3 steps: pre-fertilization events, fertilization and post-fertilization. Fertilization, as you know, is the fusion of the male and female gamete. So all the events before this are called as pre-fertilization events, and once the fusion is taking place, events after that are called as post-fertilization. In this session, we will discuss about the pre-fertilization events. Pre-fertilization events are further classified into step number 1, that is gametogenesis, and step number 2, that is gamete transfer. If you look at the reproductive part of a plant, which is the flower, so let us start the discussion with the structure of a flower. A flower consists of 4 main parts, that is the green sepals, the colorful petals, then we have the androecium and the gynoecium. If you look at the diagram of flower, these are arranged in different whorls. The function of sepals is protective. It will protect the flower bud during its early stages. Petals, you know, are large, colorful, sometimes fragrant also. Their main function is to attract the pollinators. Androecium is the male reproductive structure in a flower. Gynoecium is the female reproductive structure. The androecium, which is the male reproductive part, is represented by a structure known as the stamen. In different plants, the number of stamens will vary, and their length and size will also vary depending on the plant species. Not only that, these stamens can either be attached to the petal or they could be attached to the thalamus using a long, slender, stalk-like structure called as the filament. So, stamen is made up of a filament, and on the tip of the filament we see what is called as an anther. The end portion is attached either to the petals or to the thalamus. The anther, if you look at it carefully, it has a longitudinal line through it, and the anther has 2 lobes. Therefore, we can call it as a bilobed anther, one on each side of the filament. So there are 2 lobes, which is called as bilobed anther. Now each lobe is further dissected by a longitudinal line, which can be clearly seen under the microscope. In each lobe, at the corners, there are what are called as theca. So one lobe has 2 compartment like structures called as theca. So two theca in one lobe, another two here. Therefore, the condition can be called as dithecous. Each lobe contains two compartments called as theca. At the corner of each theca, there are what are called as Microsporangium. In total, there are 4 Microsporangiums, 2 in this lobe, 2 in this lobe. Therefore, this condition is what is called as tetrasporangiate condition. When you take a transverse section of anther, a TS of a anther, what you see is a structure like this. So these are the 2 Microsporangium and 2 in another lobe in the transverse section, and these sac-like structures called as Microsporangium, they are present in the entire length of the anther. Now, if you look at the TS of a anther, it looks circular in outline. There are 4 layers in a anther. The outermost layer is a single-celled thick and called as the epidermis. The function of this layer is protection of the inner layer, so it is protective in function. Below the epidermis, there is another layer of single layer of cells which are called as the endothecium to protect the inner layers. Other than that, the endothecium layer is hygroscopic. So when this layer loses water, it will shrink and it will help in the dehiscence of anthers. So other than the protective function, endothecium also helps in dehiscence of anther. Innermost to the endothecium layer, there are 2 to 3 layered cells called as the middle layers. Inside the middle layers can have some amount of nutrition, mostly they are protective in function. The inside to the middle layer, we have a specialized layer of cells called as the tapatum. Cells are quite large. They have dense cytoplasm which is rich in nutrient content. They store the food, and more often than not, they have more than one nucleus. They are multinucleate. Function of tapatum, because it is rich in nutrients, it will provide nourishment to the growing pollen grains. Inside the tapatum layer, when the anther is young during the early stages, it contains a mass of cells called as the sporogenous tissue. When the flower matures, the sporogenous tissue is the one that produces spores. That is, in this case, being the male reproductive structure, it will produce microspores, which later become the pollen grain. So, pollen grains are produced by the sporogenous tissue. So, this is how the transverse section of anther looks like under a microscope. Microsporangium, they are connected by a connective tissue, that is this tissue here, connective tissue. In the future, when pollen grains are present inside this, they are called as pollen sacs. Let us now discuss about gametogenesis, that is How is spore produced by the sporogenous tissue? In the male reproductive part, the production of gametes is called as Microsporogenesis. Genesis means production. Production of what? Production of microspores, which are nothing but the pollen grains. So, microsporogenesis can be defined as the process where microspores are produced by the microspore mother cell or pollen mother cell by meiosis. Each cell in this tissue can undergo division. Therefore, each cell over here acts like a pollen mother cell. What would be the ploidy of the pollen mother cell and the pollen grain? Pollen mother cell will be diploid. The diploid cell will undergo meiosis to produce microspores. Microspores will be haploid. They will contain half the number of chromosomes present in the pollen mother cell. Let us discuss a little more about this microsporogenesis. So, all the cells, the pollen mother cells, they will obtain nutrition from the tapatum and they will considerably increase in size. Later, the pollen mother cell, as you already know, will undergo meiosis. Each pollen mother cell will produce what is called as a tetrad of microspores. This tetrad, they are held by what is called as a callus, a substance called as callus. So even till here they are called, when the four cells in a tetrad separate, then we call them as pollen grain. So pollen mother cell produces a pollen grain. Pollen grain is also called as the male gametophyte. That is, it's the male body which produces the gamete. Pollen grain itself is not the male gamete. Pollen grain, it produces the male gametes. Once the sporogenous tissue produces pollen grains, that is here, diagram you can see, the entire sac-like structure will be filled of pollen grains. Now, it is called as pollen sacs. Sacs that contain pollen grains. What does the pollen grain look like? What does it have inside it? And whether there are any uses for the pollen grain? You would find that the pollen grain is usually spherical, around 25 to 50 micrometer in size. So it is roughly spherical. The pollen grain is made up of an outer wall which contains two layers. Outer layer is called as exine and the inner layer is known as intine. Inside the intine layer, the cytoplasm is surrounded by a plasma membrane. Once again, there is a plasma membrane. Inside the cytoplasm, nucleus will undergo mitosis. It is single pollen grain. The nucleus will undergo mitosis and produces two cells. So there are two cells. The larger cell is called as the vegetative cell, or it's also called as the tube cell. dense cytoplasm, bigger in size, irregular nucleus, it is filled with nutrients. Whereas the lower cell is called as a generative cell. It is smaller in size and it is spindle shaped. So this is the general structure of a pollen grain. The outermost layer, exine, it is made up of a very tough substance called as sporopollenin. This sporopollenin is one of the hardest substances known. It cannot be degraded by acids, bases or any of the known enzymes. It is very resistant to all the extreme environmental conditions. Because of this exine layer, the inner contents remain safe, and because of the presence of sporopollenin, these pollen grains can be preserved for literally millions of years as fossils. In the exine layer, there are some pores where sporopollenin is absent. These regions are called as the germ pores. The inner layer, intine, it is a continuous layer. It is made up of pectin and cellulose. This intine layer inside has the cytoplasm, which contains a bigger vegetative cell and a smaller generative cell. So this is what is called as the two-celled stage. So during gamete transfer, when the pollen grains are released from the anther, most of the plants, almost 60 to 70% of the plant kingdom, they release the pollen grain in the two-celled stage with a vegetative cell and a generative cell. The rest of the species, approximately 30% of the plants, which includes mostly the monocots, they release the pollen grain in a three-celled stage. So where does the third cell come from? The third cell comes from the division of the generative cell. It will generate, it will produce new cells. This generative cell will undergo division and it will produce two nucleus. These are the two male gametes, which is seen in monocots. In 60% or 70% of the plants, pollen grain is released in the two-celled stage. That is about the structure of the pollen grain. For example, some people are allergic to pollen grains. When you take a walk in the forest or near the grasses, people develop allergy. This is because of pollen grains. Pollen grains, they are rich in nutrition. Pollen tablets are used as food supplements. Not only that, the pollen grains are consumed as tablets or syrups by athletes, or it is given to race horses because it is known to increase the stamina and performance. So these are the two uses of pollen grains. Not only that, because the outermost layer sporopollenin is non-degradable, the pollen grains can be stored for a long duration of time under liquid nitrogen, minus 196 degrees Celsius. Pollen grains can be stored in that. These pollen grains can be used as pollen banks, which can be used in our artificial breeding experiments. Which leads us to the last topic here, that is pollen viability. Viability means how long is it going to remain active, meaning how long will the pollen grain be able to produce a pollen tube. Some pollen grains, for example in cereals like rice, wheat, they have a viability of roughly 30 minutes. After 30 minutes, the pollen grains will not generate, they will not produce pollen tube. Whereas some other pollen grains belonging to the family Leguminosae, Solanaceae, Liliaceae, their pollen grains can be literally viable for months together. That is about the male reproductive structure, that is the stamen. To quickly summarize, stamens contain filament and anther. Anthers contain two lobes. In each lobe there are two theca, and in each theca corner there are two sac-like structures called as Microsporangium. The Microsporangium or the anther has four walls: epidermis, endothelium, middle layers, and tapatum. Inside the tapatum we have a mass of undifferentiated cells called as the sporogenous tissue. When plants undergo maturity, each cell in the sporogenous tissue, it acts as a pollen mother cell. It undergoes meiosis to produce a tetrad of microspores. These microspores then separate and produce pollen grains. Microsporangium in this condition is called as a pollen sac. There is a exine layer, intine layer. In the cytoplasm, there is a big cell called as a vegetative cell and a lower small cell called as a generative cell. Most of the plants release the pollen grains in this two-celled stage. The anther, as I told you, there is a line of dehiscence. Here there is a line that runs along. When they lose water, it will shrink. When it shrinks, the anther opens and it releases the pollen grain out through this line of dehiscence. So, male gametes are present in the pollen grain, which is called as the male gametophyte. Next, we will discuss about the female reproductive structure, that is the pistil, and what events happen in the pistil.